Hi, this is Haider Abbas. I'm working as Applied Research Specialist for Manitoba Agriculture, working at Manitoba Crop Diversification Centre. Through this channel today, I'm providing some agronomic recommendations to achieve maximum biomass for the mustard biofumigation process, which is used to control verticillium wilt in potatoes. Biofumigation is a strategy which uses plants that produce chemicals called glucosinolates. These glucosinolates are broken down by an enzyme to release various substances including isothiocyanates. Isothiocyanates are known to suppress numerous pest and diseases organisms in potatoes. Since 2019, MCDC has been conducting experiments to characterize agronomic practices to achieve maximum biomass for the mustard biofumigation process. To do this process right way, certain parameters are taken into account. I will walk us through all those parameters to ensure the successful process of mustard biofumigation in potatoes. First of all is the seeding date. The research at MCDC has shown that August planting date with October biofumigation can be used to raise a successful crop provided there is not a wet fall with extended potato harvest. However, in Manitoba conditions, early June planting date with late August biofumigation has shown successful results. The reason is that early June flea beetle damage is expected to be less. In growing mustard, main goal is to have true leaves up before flea beetles begin to feed. Crucifer flea beetles thought to be more damaging than stripped flea beetles in fields. The challenge with late August biofumigation date is it is very close to potato harvesting time in Manitoba. Experiments have shown that mustard grown from June to August is less as compared to August to October, but both planting dates, June and August, can achieve 5 feet tall plant height, which is a target. Seed treatment is also one of the important factor, and it is needed for flea beetle protection. Planting method is also observed another important factor. Direct seeding is preferred to broadcasting. When we do direct seeding rate should be kept between 6 to 10 pounds per acre. Seeding can be done in both heavy and light residue situations. Direct seeding provides good seed to soil contact. When we do broadcasting, on the other hand, seeding rate should be between 8 to 12 pounds per acre. After broadcasting, a harrow pass will be required to ensure better seed to soil contact. Fertilizer application is also one of the important factors. Nitrogen is important to the production of biomass, and sulfur is crucial for the production of glucosinolates. Nitrogen is applied depending on the field's history. The rate of sulfur should be adjusted in relation to the chosen nitrogen rate in 6 to 1 ratio. For example, if 100 pounds per acre of nitrogen is applied, then the suggested amount of sulfur to be applied would be 17 pounds per acre. For the best results, pH of the soil should be above 5.5. If the field has a pH lower than 5.5, the biofumigation process might not be successful. For optimal results, the pH of soil should be as close to 7 as possible. Another important factor is mustard variety. Mustard comes in many varieties, but not all equally as effective when it comes to biofumigation. Some mustard varieties produce more glucosinolate compared to others. In fact, some varieties have been bred for the sole purpose of biofumigation. Cliente variety grows quickly and is typically used in spring or late summer, bred specifically for the biofumigation purpose as it contains very high levels of glucosinolate. At MCDC, we are testing Cliente, Cutlass, Andente, AC Vulcan, and brown mustard. Mustard crop should be incorporated into the soil before it has reached to full bloom. Incorporation process should be done when soil has a good level of moisture, and do not incorporate mustard when soil is dry. Mustard must be incorporated immediately after mowing. 80% of the fumigant gas will be released in the first 20 minutes after mowing. After incorporation, the field should be rolled and packed to trap the biofumigant gas in the soil. Finally, when the incorporation process is complete, leave the field undisturbed for 14 days to ensure all the plant's material break down. For more information and project results, please visit mbdiversificationcenters.ca or mbpotatoresearch.ca. Follow us on Twitter handle at Crop Centers. I have Zach Frederick here to provide an update on Producer's Field component of this study. Over to you, Zach.
Thanks, Hader. My name is Zach Frederick, and I am the Applied Potato Research Agronomist for the Manitoba Horticulture Productivity Enhancement Centre, or MPEC Inc. We like our acronyms around here. And Hader has already covered the agronomic component of our mustard biofumigation study and why it is so critical to focus on such a narrow set of recommendations. I wanted to elaborate a little bit on what we've been working on courtesy of grower collaborators, which is an important component of actually taking to the tail end of the experiment and determining how we finish this and treat Verticillium wilt of potato on a large field scale and making sure that it's agronomically feasible. So we've worked with growers from a couple of different growing areas, not just Carberry, Manitoba, mostly looking at lighter texture soils and fields that have been previously identified through our research as having a major verticillium problem, and a handful of new fields that are only just recently having emergent verticillium outbreaks. And so we have been working with a collaborator to have a qPCR test to quantify verticillium in the top 10 centimeters of soil. From there, what we're trying to do is evaluate paired plot situations in each of these grower fields where we have 4 negative controls and 4 mustard biofumigated plots. Everything is geo-referenced down to approximately 6 centimeter accuracy. And what we're trying to do is to resample areas that are treated with mustard and areas that are not treated with mustard, and hopefully start out with similar levels and populations of verticillium and similar levels of disease expression and on russet burbanks, and then follow with the grower as they move through several different mustard biofumigation cycles, whether or not we're seeing a decrease in the level and expression of disease and verticillium wilt, both in terms of percentage chlorosis and severity, as well as the negative impacts on the tuber size profile and ultimately the bottom line that the grower sees. So we're looking at the pathogen, the disease level component and the pathogen component, where we're quantifying the verticillium levels in the soil. And there is those that have worked with it know there's variability, highs and lows in verticillium testing. So we're attempting to quantify with that variation that occurs naturally and see overall a downward trend through the use of these mustard products when they achieve the biomass that we're shooting for through these target recommendations. And that's where this will be really tying it all together as far as whether this practice is actually dollars to donuts, making the grower money for the time invested to it, and how it compares to other verticillium management practices so the grower can evaluate which is the most appropriate in their farm setting. And we'll have more updates on our website this year at mbpotatoresearch.ca with how these case studies are progressing. Thank you.